We were going to start our international webinar on behalf of Probir Chatterjee Research Foundation and Department of Botany, University of Calcutta. So it is a great moment for us as we are organizing 20th P. Chatterjee Memorial Lecture, and it will be delivered by Professor Felix Bast from Punjab Central University. And you know, this foundation was established in the year 2003 for remembering our professor and the founder of Phycology Laboratory at CU, Dr. Probir Chatterjee. He died on 1990, and after joining here as in-charge teacher of phycology in the year 2000, we student, myself, Samit, and other general, we are PhD students of Dr. Chatterjee and other general students and his colleagues, his friends, total 25 founder members established this foundation. And every year we are organizing this P. Chatterjee Memorial Lecture since 2003. So today it will be 20th P. Chatterjee Memorial Lecture. And today with us, Vice President of this foundation, Professor Shomit Ray, whom I told we were classmates, we did together PhD under Dr. Chatterjee. He was in charge teacher of phycology at Vishwabharati Shantiniketan, recently retired. Now he is engaged in other university. So I will request Professor Shomit Ray to give an introductory lecture before going to start our 20th P. Chatterjee Memorial Lecture by Dr. Felix Bast. And I will request Dr. Nilufar Khatun and Dr. Gaur Gopal Shatpati, who are joint secretaries of this webinar, to take the in charge of this webinar and to regulate the whole day program from now. Dr. Nilufar Khatun.
Thank you, ma'am. So before the inaugural speech from our Vice President, I would like to welcome you all with a welcome song by Shrimanti Banerjee. So let's have that welcome song by Shrimanti Banerjee. Thank you so much. Now, moving on, I would request our Vice President of PCRF, Professor Shomit Ray, to deliver the inaugural speech.
Good morning, everybody. This is Professor Shomit Rai. I have the pleasant duty to welcome everybody to this Phycology Webinar. This is an e-conference which is being organized by Probir Chatterjee Research Foundation and Department of Botany, Calcutta University. I introduce myself as Professor of Botany and also the Vice President of Probir Chatterjee Research Foundation. Probir Chatterjee Research Foundation, which was established in the year 2003 with the prime objective of augmenting teaching and research in botany in general and in the field of cryptogamic botany in particular. Late Dr. Probir Chatterjee, under whom several of us have completed our PhD, mainly on the topics on phycology and a few on the bryology, was an illustrious, devoted, motivated teacher of botany. His immense knowledge in all the spheres of botany made him an attractive and very dependable personality to all the members of botany fraternity in the state of West Bengal and also in many parts of India. We have learned so many things from him, not only theoretical knowledge but also practical training in the field of phycology and also in the field of bryology. As a teacher, he had the distinction of mastering many spheres of botany, particularly phycology, bryology, teratology, and also he had sound knowledge of the angiosperm taxonomy. Simply because of the fact that he started his career in the prestigious and one of the finest college of India, Presidency College of Calcutta, and there he had to teach many aspects of botany. Ultimately, he took up research in the field of cytotaxonomy of the angiosperm family Caryophyllaceae and the algal group Charophytes. After he became a faculty member of Calcutta University, many of us joined his laboratory to complete our PhD degree. And gradually the laboratory developed. After the sad demise of Dr. Chatterjee, under very unfortunate situation, we, some of his students and some of his colleagues, we decided to form a research foundation in his name with the prime objective of carrying out programs to augment teaching and research in the field of cryptogamic botany, both theoretical as well as practical training. Our journey started with donations from individual personalities, and later on we found out some other avenues so that we could carry forward various activities of the foundation. We started with holding Probir Chatterjee memorial lectures every year, and later on we added to that organizing workshops following University Grants Commission directives, and we received overwhelming response from research scholars and particularly from young teachers who, after completion of their MSc and PhD, joined various colleges and universities where they had to undertake theoretical and practical classes on various groups of plants. And our journey continued. We organized these workshops in various universities of West Bengal as well as in Orissa and Assam. All eminent scientists from different Indian universities who were working in various cryptogamic groups, they participated, they delivered lectures, they carried out hands-on trainings, and that was a big success for this research foundation. The students, research scholars and young teachers were very much benefited, and they showered their appreciation on us, and that was our real success. Subsequently, we have taken up publishing an e-journal, and also we have our YouTube link where we are loading various lectures both on theory and practice. For last few years, we are organizing e-conferences. We were somewhat forced to do that because of the pandemic, and here also we received overwhelming responses from India and abroad. This e-conference is one such endeavor on behalf of Probir Chatterjee Research Foundation and Department of Botany, Calcutta University. We are grateful to the university administration as a whole and the Department of Botany administration led by Professor Rita Kundu as head of the department, who have extended all kinds of facilities and assistance so that we could organize this program smoothly. So with this few words regarding the Probir Chatterjee Research Foundation, its activities, and the contribution of various academics, particularly Professor Ruma Pal, who as the founder general secretary of this organization have single-handedly carried many responsibilities to make it a fruitful event. So with these words, I invite Dr. Felix Bast, Professor and Head, Central University of Punjab, Dr. Andrzej Witkowski, Professor, University of Szczecin, Poland, and Professor B.B. Chaugule, formerly Professor and Head of the Department of Botany, University of Pune. Sir, your agreeing to deliver lectures for our participants and to make the efforts of Probir Chatterjee Research Foundation all successful is gratefully acknowledged. We are deeply indebted to you that you have agreed to deliver lectures, and it will fulfill our efforts, our dreams, so that our students, the participants of this workshop, can enrich themselves with the state-of-the-art knowledge that you will be sharing with them. Thank you so much. And with this, the e-conference is open for delivering of the lectures. Thank you so much.
Thank you, sir. Thank you so much. So moving on to the 20th P. Chatterjee Memorial Lecture, which will be delivered by Professor Felix Bast. Sir, we welcome you. So before your lecture, Professor Felix Bast is a professor in the Department of Botany, Central University of Punjab. He is an expert panelist of Paris-based International Science Council and elected fellow of Linnean Society of London and a member of IUCN Geneva. He holds PhD in marine biology from MEX, Japan, and served as expedition scientist in Indian Antarctic Mission, international doctoralship, and also he discovered 7 new marine algae and moss species from India and Antarctica. He is an alumnus of IIT Bombay, University of Washington, US, Marine Biological Association of UK, Plymouth, UK, and Leibniz Institute of Tropical Marine Research, Berlin, Germany. Professor Bast served as an in-residence intern with the President of India at Rashtrapati Bhavan, New Delhi, and received President's Inspired Teacher Recognition in 2015. He also won the Teaching Innovator Award from the Ministry of Education, Government of India in 2019. For his research and dissemination, he had been to 25 countries. He also popularizes science through several media. He has founded Young Academy of India, an academy for young researchers in the country, which is member of International Science Council. He has published more than 65 research articles in Scopus enlisted journals and guided 7 PhDs, 2 postdocs, and 1 international exchange scientist. His works include Life Skills, Voyage to Antarctica, and Textbook of Biostatistics and Mathematical Biology. He advocates a sustainable lifestyle, a primary focus of his work. He's also an avid cyclist who often commutes to his workplace on cycle. We welcome you, sir. Please, over to you.
Yeah, thank you so much, Professor Rumapal, for having me here, for inviting this very prestigious event and this particular talk on remembering on Professor Probir Chatterjee and his contribution to the field of phycology. Let me share my screen. I have a small presentation for you. So the title of this talk is Curious World of Algae. I hope my voice is clear and audible to all of you. Can someone
Yes, sir.
confirm?
Yes, sir.
Yes.
Yes, sir.
Yes, so in case my internet fails, I have already shared this PPT to the organizers. So you may please, flash this slide so that I can try to join by my mobile phone. Okay. So sometimes it doesn't work here in my workplace. So I'm talking to you from Central University of Punjab. It's a new university established in 2009. And we are a NAC A graded and NAR of top university among Central universities, the newly established Central universities. And we are located in a small village called Calcutta in Bathinda district. Again, it's one of the remotest locations. So, you know, it's very near to the Pakistan border. I don't know how many of you have been to in this area. So here I work, mostly my work is on algae and especially marine algae. And you might wonder, how do I get the samples? So of course I travel and I send my students as well. And I also go for exploration, and explorative work fascinates me a lot. So thanks a lot for this particular invitation. I'm really honored to spend the next an hour or so with all of you. And so Probir Chatterjee's contribution is profound. I will come to it in a short while, especially on as the professor in Department of Botany said, on Cara and the taxonomy of the Cara, under whom Professor Rumapal, I understand, did her PhD as well. So coming to this particular lecture, this talk is really general, and this is just to fascinate you to the exciting world of algae and why the algae matters. Right, and today is also coincidentally a very important festival in the state where I'm from. I'm from Kerala, and today is a festival called Onam. It's a festival of flowers, you know, and plants and harvest. Right. So, and also coincidentally, again, today is the birthday of my mother. Right. So it's a lot of interesting coincidences. If I ask you to close your eyes and think of planet's natural oxygen factory for a moment, what image comes to your mind? I'm sure the image is going to be something like this, a forest canopy full of trees, because that is what we have been taught, right? The oxygen producing factories are trees and plants that we see in the terrestrial. What if I tell you it's a myth? As we all know, 71% of the planet's surface is water, and the water has, if you zoom into any of this droplet, the oceanic droplet, you can see tremendous diversity of phycoplanktons, the floating unicellular bacteria, right? Of course, these are also part of algae, and these are all photosynthetic phycoplanktons. As the introducer said about me, I'm also into science communication, and several of my articles on algae have been published in popular journals, including Dream 2047 and Science Reporter and Resonance. Please check it out. This is one of the articles which got so much of the, so much of reading I got. Still, I get, the emails about this particular article. So 2 key living organisms that may save the earth. So what are these 2 key organisms? Synechococcus and Prochlorococcus, just 2 species of phycoplanktons. And why it matters a lot? Because it produces over 65% of the oxygen in the air that we breathe. Isn't it exciting? And it is something which we completely forget, even the mere existence of these phycoplanktons in the world's oceans, and it's completely forgotten in school syllabus of the country. And not just here in India, but anywhere in the world. Usually we don't speak about these organisms, albeit it's tremendous importance. So algae is extremely important, and they were the first living organisms, the first, I can say, the plants which ever came into existence, right? Just after the RNA world, as you can see, the origin of life, just soon after the origin of life in Archean, the cyanobacteria appeared, right? And from Archean all the way to the Phanerozoic, just before the Phanerozoic is the Ediacaran, when the multicellular organisms start. So the algae ruled the world, and even the first eukaryotes were seaweeds. As evidence say, it is red seaweed used to be the first, the oldest seaweed fossil ever recorded is a red seaweed. So stromatolites, as you know, it's the oldest fossil ever known. It is nothing but biofilms of the cyanobacteria, approximately 3.5 billion years old. The oldest fossil record is with the bacteria. It's an algae, it's a cyanobacteria. And can you guess what is the smell of the dead body like? The dead body of this, the algae. So to know that the smell of its dead body, all you need to do is to go to the nearest petrol pump. Yes, the petrol, the gasoline. 99% of the petroleum comes from the biomass of the marine algae. Another curious fact that most of us don't appreciate. Most of these are diatoms and coccolithophores. As you know, coccolithophores, so beautiful creatures, like as you can see in this picture, Emiliana huxley, belongs to Chrome alveolata. It's a group of, the brown algae. Chrome alveolata is, as per the latest systematics, Chromista and alveolata together, right? Alveolata is having small air-filled sacs like dinoflagellate, apicomplex, and ciliates and all. While Chromista contains haptophytes, cryptophytes, and straminophiles. So straminophiles are usually our diatoms and brown algae, and also golden brown algae, chrysophyceae. So coccolithophores belong to haptophytes. And I was privileged to be part of the Indian Antarctic mission. And in Antarctica also, I've seen so many interesting kinds of algae. For example, this picture, which I took in a lake, it's an oligotrophic lake, glacial meltwater lake with almost no nutrients at all. And still you can see froth, so the froth reminds me of my time in Delhi. If you have been to Yamuna, the banks of Yamuna, a lot of froth comes, right? It's our industrial pollution. But in pristine landscape like in, Antarctica, how can you see this kind of froth? It perplexed me, and I analyzed this one, and I confirmed it is Phycocystis antarctica. It's another brown algae. So the phycocystis, it itself is an adaptation for flying. When you go to this particular lake, you can see this frog flying around. So it can actually transport by the wind from one lake to another lake. Fantastic adaptation, isn't it? And how many of you know about the curious connection with the algae and monsoon rains that India gets? So the tiny ocean plants can help make the clouds. So these are cloud seeding molecules, the DMS, dimethyl sulfide. So dimethyl sulfide gets released by phytoplanktons, and especially this, the coccolithophores. So, and there are interesting studies that these coccolithophores, when it get affected by marine viruses, the viral infection, it gets dead, right? So that actually, that mass bloom followed by its death releases tremendous amount of dimethyl sulfide, which escapes to the clouds, and that seeds to form the monsoon rain clouds. So very interesting studies are revealing these days. So another of my interesting work is about the blood rain. So blood rain, the phenomenon has been reported since time immemorial. If you look in the Bible, you can see instances of the blood rain. And even in Homer, the Greek, the writer, the ancient writer, his Iliad and Odyssey has these instances of the blood rain. And in South India and Sri Lanka, we get this blood rain almost every year. Some parts of the Kerala, we get it. And of course, there were so many theories why this rain, blood rain means it's just red colored rain, that if you put the clothes like I'm wearing today, the white cloth, if you put for sun drying, and after this rain, the white clothes turn into red, and why this happened. And you can see that this one of this paper cutting is about the unexplained theories, the extraterrestrial theories on alien rain. It's because of the aliens. So we collected the samples and we did analysis, and it turns out to be a green algae. Trentipolia annulata is it's a green algae of Trentipoliales of the Ulvophyceae. And this one is a spore dispersal mechanism of this algae. The sori of this algae gets released and it goes to the cloud, and then it disperses to a vast area. Fantastic adaptation, right? Just like this milkweed, you can see that in very common in South India, and I'm sure it is there in West Bengal as well. Asclepias eriocarpa, right? And also Gossypium, the cotton, right? So yes, so all these are the wind adaptation for its pollination and seed dispersal. Same kind of adaptation is what we discovered. Our team from Central University of India discovered way back in 2014, unraveling the blood rain history. We did the DNA barcoding. So the kind of work which we do combines the exploration and morphology-based field identification combined with the molecular work, including biochemistry and also DNA barcoding to facilitate its the species delineation and species discovery. It's another very interesting paper which I read recently, and I do have a YouTube channel. If you're interested, please check it out. It's Felix Bast. I have around 13,000 subscribers. And in my channel, I do have a science program every month. It's called Curiosity. So in one of the episodes, I featured this particular, the paper, snow meltdown in Himalayas accelerate nocticular algal blooms in Arabian Sea. That's fantastic discovery, isn't it? So it's very curious fact how the Himalayas snow meltdown is connected with algal blooms in Arabian Sea here in Sindhudurg district of Maharashtra. So nocticula, by the way, it's actually a bioluminescent algae. Okay, so that's pretty interesting connection with the global warming and increased incidences of such algal blooms throughout the Arabian Sea, including Omani's coast and Iran and Pakistan and Gujarat, all the way to the Maharashtra. The incidences are increasing. If you have seen Life of Pi, the movie has a scene of this bioluminescence of this nocticular scintillant cells, and of course the dinoflagellate has several health implications as well, especially shellfish toxin. Several of these dinoflagellates are toxic, the neurotoxin. Carenia brevis has got the brevitoxin, the red tide, right? You can see that this is a huge expanses of the ocean. This is just a container ship. You see that shipping liner, such a big ship is just a small fraction, just to give you an idea about how big is this image, right? And this entire area is now infested with the brevis, Carenia brevis. And also you can see this particular thing as well. So massive red tides are usually caused by the dinoflagellate, and brevitoxin is a neurotoxin, right? And especially if you consume the shellfish and oyster, could be potentially lethal, right? And also gymnoidium is another dinoflagellate, gymnoidium, and of course the goniolax as well. Goniolax is also the reason for this red tide, right? And most are toxic red tides. And another very interesting paper which I featured in my program is coming from this Namibia and Botswana. So lakes in Namibia and Botswana, nearby the lakes, a satellite picture shown so many elephants dead. And the phycologists did the assessment of those lakes to detect. So you can see the carcass of an elephant just near this lake. Yes, it's because of the algae, infested with algae like Physteria, another dinoflagellate. Physteria causes mass death of fish. It's a fish poison algae. So algae is, it's as usual with any plants. Many are toxic, but many are edible as well. And many have potential therapeutic agents, like even for the COVID-19, there are several such studies have shown that the complex polysaccharides, sulfated polysaccharides, like fucoidans extracted from the seaweed Saccharina japonica, have shown anti-SARS-CoV-2 actions, in the cell lines. But as you know, the problem with most of this bioactivity assessment is that it works in cell line, but it doesn't work in clinic, right? So that scale up is a major constraint with most of these bioactivity studies, and also how clinically significant is this one. And another very interesting paper recently published is about in Science Advances, a very prestigious journal. A team from Cambridge University, artificial regulation of state transition for augmenting plant photosynthesis using synthetic light harvesting polymer materials. So they are boosting the photosynthetic rate of algae like chlorella. Why you need to boost it? Because right now itself, the chlorella has the best photosynthetic efficiency of any non-plant, but you can still boost it by applying the principles from the quantum physics. You can boost it for, yes, to make biofuel from it, and also for carbon capture and sequestration to remove the carbon dioxide from the atmosphere. So yes, you can see that Inverse ran a story on, it could mean cars running on algae. I mean, the stories like this comes again and again, but unfortunately till date, nothing substantial have happened, but hopefully in the near future, something interesting will come up. And there's so many interesting aspects of the algae that most of us are never known. I'm not sure how many of you knew that algae is used in medical forensics, including a rape case, a very famous rape case in Shopian district. It is a purported rape case that the locals blamed it as a rape. But then what happened is that, a diatomologist she's working in Chaugule University right now. She was earlier with the forensics department. Did very interesting assessment on the lung to see that the cause of death is, drowning. It's not the rape. So you can see that this kind of thing. So is it really somebody killed and put into, into a water body, or is it really drowning? So if you assess the tissues from the lungs, you can see it. And also if the death happened in one lake and then took the body and put into another lake, that also you can find by looking what kinds of diatoms are there in the lungs. So very interesting utility of this algae in the forensics. And of course, the medicines from algae, marine natural products, several, chemotherapeutic drugs as well available in the market. Taxol, which has a root onto the Plaxitaxel is the brand name from Bryopsis plumosa, which is nothing but an algae. And many algae are edible and very expensive. So some of the algae, and the algae which I worked for my PhD is on Monostroma cruicense, the one which I named it. It's the second most expensive algae in Japan, where I did my PhD. So algae are very expensive. If you search in Amazon, you can see Porphyra sheets, just like 50 grams will cost you around 600 rupees. And why you want to, import this algae from abroad? We have excellent resources of algae in our cost, but unfortunately, we don't have much industries, and that is why Government of India has now a new policy called Blue Economy that envisions more and more industries, seaweed-based industries. Okay, so mariculture, that is also part the algae farming, right? Algae is also part of the mariculture. Beyond algae, of course, the shellfish industry is also part of the mariculture and also fish, right? Aquaculture. The beauty of this algal farming is that it needs no fresh water, no land, no real estate is required, and of course, no fertilizer, nothing is required, just ocean. That we have a plenty, around say 7,000 kilometers of coastline India has. And it's pitted as the sustainable agriculture for the to ensure the food security of the future. And do you know how many of you know the flamingos? Of course, you can see the flamingos, the birds and migratory birds in Kutch district in Gujarat. You can find seasonally the flamingos. They're pink because of algae. The chicks, the flamingo chicks don't have any color, and they eat algae as a food, and also the fish which eats algae, and it gets this pigment from the carotenoid produced by the algae. Exciting world, right? And such very interesting, curious facts. I recently developed a science show for Vigyan Prasar and Prasar Bharati. The science show is known as Mysteries in Science, 10-episode series, which will soon be aired in Doordarshan. Please wait for it. Okay, so it's a major project which we won around 30 lakhs, and everything is completed. The recording is completed in the studio, and also outdoor shootings, right, in Delhi and elsewhere. Another very interesting, curious fact is a sloth. The one usually they hung in the tree. It's an arboreal animal. It forms algae on its body. How exciting is that, right? Algae, the sloth comes down to defecate, and during that process, it gets the algae on its, on its fur, and it eats it. But when you lick your fur, its fur, it gets the algae as the food. How exciting is this kind of curious research? So if you're a youngster, if you're a student, check out for exciting world of algae. Algae is not just, the lab work. The field is a lot more exciting for you. So many interesting facts. Prime Minister made a statement that the farming of seaweeds can enhance water resources. Yes, we have a draft policy on the blue economy by Ministry of Earth Sciences. It's put up for public opinion, and you can comment on it. It hasn't been ratified yet, but farming of seaweed is pitted as the sustainable agriculture of the future. This is an algae which I worked on almost 5 years in Japan, Monostroma cruicense of the Kuroshio current, and this is how they cultivate the algae. And this is used for sushi nori, sushi wraps, just like the nori. Nori is basically Porphyra, but Porphyra is quite expensive red seaweed, and this is the second most expensive seaweed in the world. Monostroma cruicense, the edible algae. Right, this is of course the green seaweed of the Ulvophyceae, and as you can see that these are raft method of seaweed cultivation, and we have several such method for the offshore cultivation. For example, the first one you can see it's a floating raft. It keeps on floating. 2 levels you can see the water level. One is a high tide and this is low tide. Second one is semi-floating raft method. Again, during the high tide, it's floated so that the algae is actually exposed to the sunlight. While in the low tide, it's completely exposed so that it gets a chance to dry up. The first method, algae doesn't get dried. It's good for Saccharina, Undaria, Eucuma, Kappa ficus, and Sargassum. While the second method is good for Porphyra, Monstrum, Ulva, and so on. And the third method here is off-bottom shallow water. This is exactly what you can see in this figure, off-bottom shallow water. The nets is completely immersed in high tide, while completely exposed in low tide. It's good for monostroma and porphyra. While the fourth method is the bottom planting, Gracilaria and Caulerpa, this kind of benthic algae, right? Can be cultivated by this method. So what is our contribution to the field? Our means my group here in Central University of Punjab. The main contribution is that we discovered 7 new species known to the science. These are all the isotype and the holotypes for you, because you're from Calcutta, it's very easy. You can just go to the Central National Herbarium. All the holotypes have been archived forever in the curatorialship of this Central National Herbarium in Calcutta. So 6 are the new species of marine algae, the seaweeds from Indian coastline, while 1 is a small species from Antarctica. These are the species, right? And this one is the Bryum Bharatiense, named after Bharati Station and also the country. It's from East Antarctica, right? And of course, among the algae, 4 are green, while 2 are, it's a hypnea, it's a red algae. So Ulva Paschima, the name, so because of the western coastline, I published this one in +1. Cladophora goensis is because of the Goa, the type locality. Ulva uniseriata, Bast, is actually coming from 2 locations. One is the Diamond Harbor, the south of Kolkata, right, in West Bengal. And that's a very interesting algae, which I found. Some of my work have been featured in BBC. It came into the front page. The first report is India discovers new plant species in Antarctica, right? Bryum Bharatiense. And the latest species which we discovered is called Acetabularia jalakanyakai. Earlier, it thought to be just Acetabularia acetabulum from Andaman, but our molecular work revealed that it is actually a new species. The little mermaid, jalakanyakai, is like the Sanskrit of the mermaid. I was privileged to be part of this Indian Antarctic mission, 36th mission, that is in 2016, December till 2017, April. And I got a golden chance to do the research near Bharati Station in Larsman Hills and Maitri Station in Shirmakar Oasis. So I completed the work in these 2 locations. I surveyed for interesting, phycological diversity. I published a book called Voyage to Antarctica, which is rather a popular science book, which, in addition to the popular aspect of the Antarctica, enlists my what kind of research which I did. Pink ice algae I found in Antarctica. That is also pretty interesting. It's something like the blood rain, which I told you earlier. This is the pink color is also because of the carotenoid pigment, like beta carotene. The entire area gets bloomed because of this. So when you culture, of course, we are maintaining this in culture in the lab. It is no more pink, so that carotenoids are not expressed in the, in the comforts of the lab. So most probably these pigments are expressed in response to stress. Maybe UV light, the Antarctica has this ozone hole because of that. And Calophyllus astrosanguinea is the seaweed which we found in Antarctic coastline. So as Himantothalus grandifolius, a large brown seaweed. Mesotonium and also Prasiola crispa. We found it is in the terrestrial near the penguin poop area, right? And also we generate the floristic map of Larsman Hills region all around the Larsman Hills and all the lakes around these regions. We made this thing. Salingia subintegrator, the first report from Indian subcontinent. Salingia is again, it's an endophytic algae. So algae living inside another algae, right? So that is also a very interesting study which we found inside. As you can see here, it's actually the cladophora. Inside the cladophora, you can see the Salingia. Ulva sapora is another first report from India. Previously, sapora has been reported only from Antarctica, Australia. So our work have revealed it. Ulva shanxiensis is yet another interesting news. So earlier it was only from China, And now that we found it in India as well. Ulvella leptokeet is another interesting paper which we published. It's an endophytic algae inside the cladophora. You can see that cladophora as well as inside, other algae, so that is very interesting finding. Invasive algae also, we found several such algae like Caulerpa scalpeliformis, Caulerpa racemosa, Caulerpa taxifolia. These are invasive in Mediterranean, while in India these are not invasive. Okay, so these are native to Indian Ocean region. And one of the algae which we found is Sargassum zangii in the coast of Tamil Nadu. And of course, the zangii has been reported only from China before, so it is an invasive species. It's a report from the Hindu featured this one. Other algae from our collection include Borkensia foxii, Bryopsis plumose. I told you Bryopsis is very, very interesting algae. It has the, it has anti-cancer compound and acetabularia. Dunaliella salina is another exciting algae. It is hypersaline, hyper salt tolerant algae, and it is cultivated. You can see that in the salt paints. This is cultivated for the pigments and antioxidants, the beta carotene, right? And what you can see here is basically the salt ponds in San Francisco. I started my entire work on algae from this algae. I did a Indian Academy of Sciences summer fellowship program. Nowadays it's called SRFP, Summer Research Fellowship Program of the India. It's a very prestigious program for the young students. I mean, if you're a student, please to apply for it, right? It's a fully paid internship. I worked with Professor Matthew at National Center for Biological Sciences in Bangalore, TIFR. I worked on this algae. Chlorella vulgaris won 2 Nobel Prizes. Otto Heinrich Warburg in 1931 for cellular respiration, while Melvin Calvin in 1961 for Calvin cycle. And the efficiency is 8%. So it's also a superfood and also cultivated for protein and PUFA. And also for biofuel, we have Botryococcus brownie, high lipid content, about 40% of its dry weight. Compare that with the Jatropha. Jatropha is a land plant, oil plant, just 32%. But this is so much higher in its content. So it's a top candidate for biofuel production. Ulvella leptokeet, which I already told you about. Erythrocladia also. Agarophytes, we have a rich canopy of agarophytes in India. These are some of my picks. Dacilaria corticata, Gratilopia felicina. Caraginophytes too, for Caraginan, Eucuma, Hypnea, and Capophycus alvarazi, right? We do have endemic seaweeds of the Andaman, Dichotomeria and Galaxura rugosa, right? So these are endemic. So Galaxura is very interesting. It looks like a soft animal, but it's actually it's an algae. Porphyra, friends, we do have Porphyra in Indian coastline, in Tamil Nadu as well as in Andaman and Nicobar Island, and also in Kerala, and it's very, very expensive algae. Sargassum millicifolium as well. The pneumatocyst of the sargassum helps it to float, right? Turbinaria ornata, Padina, and Fucus diasticus, some of the algae, which you can see that sea palm and giant kelp. Unfortunately, these are not pics are from US. I was part, my PhD program is a combined one with Friday Harbor Labs. So this is a picture from Friday Harbor in Washington, in the west coast of US. Sea palm is Postelsia, and this one is actually taken from the Monterey Bay Aquarium in San Francisco. Some other famous algae, important algae is Wakame, that is Undaria pinnatifida, and Kombu, that is Saccharina japonica. It's very, if you're in Japan, I'm sure some of you have been to Japan, you can see the tea served with kombu. Kombucha is also getting popular in India, but unfortunately the kombucha, what you get here in India is just salted. It doesn't have the kombu. It should have this green seaweed to call it as a real kombucha. And these are all edible algae. Coming to freshwater algae, it's, the diversity is tremendous. Our group is, of course, working on molecular systematics of the freshwater algae from this region, the Punjab region. Chlamydomonas, volvox, and Rhizocranium, And if you look at the systematics of the plants, so Archiplastida, the crown group called, right? So chlorophytes and embryophyte, right at the root of embryophyte, entire land plants, resides this particular organism, the Chara, right? The stonewort Chara here. The featured one is the Virgata. You can see here the bright orange one, that is the antheridium of this Chara, interlocking the field cells caught just before the rupture. Very interesting picture. It's not my picture though. This picture got the British Phycological Society's prestigious Linda Award. Okay, so it's one of my favorite picture. And coming to the Chara, friends, Spirogera, and of course that is also part of the same group. You can see that if you look at the group Chloroplasta or the Viridae plantae, it's chlorophyte and streptophyte. Charophyte is part of the streptophyte group, while chlorophytes are all green algae. So streptophyte, if you look, it's charophyte and embryophyte, the entire land plant. So it is really the most ancient living, plant, the MRCA, that is most recent common ancestor for all the land plant is the Chara. And of course, the Chloroplastida and other groups include Rhodophyta, that is the red algae, and Glaucophyta, less studied algae, especially in India. Not very many people are working on Glaucophyta. So tremendous opportunities exist for young researchers who are listening to this talk. So this Chara, friends, so much of the Chara in India have been revealed by Professor Probir Chatterjee. And so this talk which I'm delivering is in his memory, in the kind of work that he did in Presidency College and also in Calcutta University in Department of Botany. So so I'm really humbled to got this opportunity to talk to you all about the algae. And also I met this gentleman, Professor Noor. I don't know how many of you know. He also did a lot of work on Chara. And he's from Uttar Pradesh College. And I met him in Madras Christian College, where I shared a room with him in the MCC guest house in one of the algal conferences. And unfortunately, within a week, he passed away, right? I'm just remembering Professor Noor's contribution to the field of Indian charophyte algae as well. And algal symbiotic systems are another exciting field. The featured one is the lichen. And lichen forming fungi is around 20,000 species, while we know only around 156 species of photobionts. The reason is very simple. People are not really working on photobiont diversity. So I still have a major grant from CSIR on the diversity of photobionts from Himalayas. Okay, so what is known, if you can see that Trendipolia and Triboxia, almost all known the green algae, the photobionts belongs to the green algae. And the quarter belongs to cyanobacteria, also Cytonema and nostoc, and the photobionts may have co-evolved with the mycobiont. That is also very interesting, co-evolution. It's a tremendous knowledge gap as well. And there is a horizontal gene transfer also. There is some new reports coming out. So my lab work includes Himalayas, especially in Kargil, Himachal Pradesh, and also in Antarctica, Larsemann Hills and Schumacher Oasis, and the connection between these two. That is also very interesting. Millions of years ago in Pangaea, of course, we were all together. Gondwana, of course, even before the splitting of Gondwana land that happened around 300 million years back, India and Antarctica were conjoining. And any guesses where in India was part of Antarctica joining? South Indian coastline, friends, and also the West Bengal coastline too, where you're listening to me. Digha, for example, you go to Digha and contemplate. Digha coastline was part of Antarctica 300 million years back. Exciting, isn't it? So any plant system that evolved more than 300 years back, you can see the similarity of India and Antarctica. That is what we did that in molecular work. So we did extract from several kinds of lichens, several kinds of photobiont, and we are maintaining in the culture. We did DNA barcoding of all these things, and we found very interesting findings of these photobionts, including Umbilicaria and Santoceria, right? So yes, so that's the very interesting work which we did, just to give a glimpse of the left and right. You can see the left side is the host, that is a mycobiont, while the right side is the photobiont, that algae. So left is fungus, right side is the algae. So you can see that the co-evolution as well. Some of these lineages have co-evolved. Another exciting field of this symbiosis is the coral reef ecosystem, the most productive system in the ocean. These are rainforest of the ocean. And coral reefs are nothing but polyps of cnidarians, which are related to the jellyfish, and the algae. So without algae, coral reefs cannot, it completely collapses, right? This is my own picture while approaching the Andaman and Nicobar Islands. So if you go to the Nicobar and Andaman, so just before landing the flight, of course, the land will land in Port Blair. If you look out, you can see that the entire area of the ocean gets this so beautiful, the color, right? So this color is all because of the reef, coral reefs, so reef building coral, and also atolls. You can see in Lakshadweep, atoll reefs are there. So yeah, one of our paper is that first report of a new kind of soft coral from India, that is Zoanthus pulchrilus. It's not the discovery of a species, but it's a new report from the region. Okay, so it's an anthozoan, and of course, no medusa stage in this algae, right? So zooxanthellae, that is basically the primary producers of the coral reef ecosystem, is an algae, Symbiodinium, which is part of the alveolata. Remember the chrome alveolata we spoke about earlier, chromistine alveolata, right? So it belongs to the SAR supergroup. The SAR means, staminopile, alveolates, and rhizarians. That is basically protozoa. SAR is the latest classification, right? All these are now part of one group. We have excellent evidences. So if you look at the pH of the world's ocean, so it used to be 8.2 for the last 300 million years. And right now it is 8.1. Or you might wonder, it's just a small, little bit decreased. But friends, 0.1 of this decrease in the pH is tremendous. The reason is very simple. pH is log scale. So in the logarithm, just 1 is, 0.1 is significant, substantial. So because of this acidification of the ocean, coral reefs are completely dying. The reason is very simple, that algae gets died, right? Symbiodinium gets died. So chrome alveolata, as you can see, the dinoflagellate is part of the alveolata. Alveolata is part of the SAR supergroup of the chrome alveolate as well, right? So it is highly susceptible to the ocean acidification. So once the coral reefs get bleached, as you can see in Lakshadweeps, so most of the coral reefs are now like this. Unfortunately, we cannot revive them. It's gone forever because of the ocean acidification. It's a bleached coral reef system. So climate change is real, friends. How do you mitigate the climate change? Again, the algae comes in picture. Carbon capture and sequestration. So removal of the carbon from the atmosphere and converting this carbon for useful energy. That is what CCC means. CCS means carbon capture and sequestration. Yes, algae does majority of the natural carbon sequestration for the last millions of years. That balance is now affected because of the fossil fuel emission, So that we have been thankful to the algae, right? Like codium, you can see. Yes, algae is the reason for the carbon capture sequestration that has been enabled us to mitigate the effect of the climate change. But now, because of that, we have to go back to the help of the algae once again, right? And as you can see, this picture is a very famous picture. This is blue marble taken by Apollo 17 in 1972, December 7th. So 45,000 kilometers away. You can see it's blue color all because of the ocean. And yes, oceans have this algae, and that is the reason why CO2 is being removed. And that is exactly what we are burning in the petrol, right? So the energy in the petrol is actually coming from algae, which remove the CO2 millions of years back. We are simply pushing it back to the atmosphere, right? So of course we can use this, we can leverage these phytoplanktons and other algae for betterment of human beings. So when you see this blue marble fence, now I hope you can now appreciate the tremendous role of algae that we have in our life. Every 2 breaths we take, friends, are coming from algae. Out of 3 breaths, out of 3 breath, 2 coming from the algae. So the planet's natural oxygen factory, the same sentence we started this entire discussion with. I'm sure now, it is, I hope you appreciate the importance of the algae now. And I hope this brief talk has been useful to you. And I'm open for any questions you have. Nandi namaskaram. Nandi means thank you so much in Malayalam, my mother tongue. And this is the link of my YouTube channel. So please check it out. I do have a program, science program, monthly science program, and that has been translated to, guess what? Your language, right? Bengali. We have a large group, Young Academy of India, and there are several volunteers that do the translation of this into major Indian languages. So every month, the Curiosities Bengali episode are also being released simultaneously. So please do check out, and thank you so much for this opportunity. And I'm really privileged and honored, Professor Rumapal. Thank you so much.
Thank you, sir. Thank you so much. It was indeed a very nice lecture. I hope everyone enjoyed. I think Professor Rumapal wants to say something, ma'am. You want to? So a little technical glitches, as you know. I think she wants to say.
Am I audible now?
Yes, ma'am, you're audible.
Okay. So just ask if there is any question
Yes.
by student, then I will say. Okay.
Now the session is open for interaction. Till now we have not got any questions, but if anyone wants to interact or have any questions, or anyone, please. No, ma'am, not yet. I think sir has provided the link and the mail. I think they can contact him directly. That's why. Maybe.
Yeah, and it was
I'm
an
also
excellent.
Happy. Yeah. I just want to add one more point here, and I'm really happy to see also a face here, Dr. Gaur. Arida, can you please unmute yourself, Dr. Gaur Gopal Satpathy? I have communicated to you earlier, right, through email. I haven't seen you. And so are you a faculty in Department of Botany? I guess you are one of the coordinators.
No,
here, right?
actually, Gaur, where are you? Gaur? Yeah, he has come.
Yeah, I don't hear you.
Unmute.
Yeah, he's unmute only. Gaur, you are not.
Now I am hearing.
Okay. Yeah, you're audible.
Yeah.
Yes.
So Professor Bast wants to know, you just introduce you, what you are doing.
I think actually, we have been communicating, right? Yeah, so I remember that many of your mails, and I think you're also part of it.
Yes. But unfortunately,
so glad to see you.
Time was actually, that time was very difficult to contact sir for regarding the work because my work actually under PhD program. And that time was very difficult, but I actually emailed you recently regarding collaboration or molecular works on
sure. I'm open for any kind of collaboration. Let me know how can I help you. Right?
Okay. I will contact you.
Actually, Gaur did his PhD on biodiesel.
Okay.
Nice. From our lab, biodiesel from algae. He was in CSR project. And then he joined as assistant professor in a graduate college in Calcutta only.
Okay, how nice.
He is trying to get some opportunity to do good research.
I'm teaching in an undergraduate college, but still doing research. So I'm interested on Sundarbans, and they are actually changing their environment according to
Nilofer, some
recent
echo
scenario.
is there.
Yes, yes. Sir, actually, we have one question over here. Topos Dash, he's asking, sir, is it possible to establish culture of all the species isolated from lichen?
Yeah, well, in our experience, most of this, the photobionts we could able to culture. So culturing, I don't think there's going to be a, the problem. But of course, you need to spend some time on standardization, So yeah, so different kinds of media like bold based media and other media, you can experiment with. And yeah, so I don't think it is really fastidious. These are nothing but unicellular green algae, so we can easily cultivate. We didn't encounter any problem with that.
Thank you, sir.
Nilofer, there is echo problem.
Hello? No, ma'am, it's
Yes.
clear. Anyone having any audible problem?
No problem? Oh,
No,
then
sir,
it's
no,
all
no,
right.
ma'am. Okay. So
Okay, is there any question from
No,
student front?
no. I think he has given a very good lecture, and he has given his mail ID and YouTube channel. So hopefully they will contact him over there.
Yeah, actually, it is really a nice opportunity for us to have Felix Bast. He mentioned about that Madras Christian College symposium. I was also there, and I met him for the first time. Both of us
Right.
actually in the
Yes.
same car when we are going to airport.
Exactly.
And I followed his lecture, and then only I thought I will bring him to my department for the sake of my students. He did a great job. All of you have heard. Most interesting, Nilofer and Gaur will remember that Gaur was not there, Nilofer was there. When we went to Crusadai Island, that's in that ecosystem, it was really for us which one is algae, which one is animal, and which one is the corals. The lower groups, they are growing, mixing. One biomass we collected, we thought it is Gracilaria. So we came with that biomass. Suddenly it started walking,
I see.
So we throw it
Interesting.
out. So in this ocean ecology, it is very tough. And as you are doing this molecular work and the isolation of pure strain from lichen and from other, it is excellent work. And I am really very happy that this type of research is going on in one part of India, because in our country there is the mega diversity, and you have actually touched all the corners. That is very good. The freshwater, then marine, and that lichen, and then these Treboxyophyces and all others, phycobiont, that is also very much needed for this molecular work. So myself and Somit, we are going to retire soon. So I must say my young students, to go to join your lab and
Yes,
to
sure.
make your hand strengthen. Somit, you want to say? And thank you again, Felix. You want to say
Thank you.
something?
This is for the first time that though virtually in contact with Professor Felix Bast, and this was an excellent lecture, and it was quite clear that he kept the students' interest in mind and not projecting high-profile research. So this will definitely motivate and encourage the students to pick up algae for their future career. Thank you so much, Professor Bast. Hope we will be meeting again in future.
Yeah. Thank you so much, everyone. It's
I
my
must
pleasure.
say
Thank
one
you.
thing.
Yes.
Thank you again. And we will actually post the memento also, this P. Chatterjee Memorial Lecture memento
Yes.
by post. Okay.
Yes.
Ma'am,
Thank
can
you
I
so
say
much,
something?
ma'am. And Dr. Gaur, yes.
Yes, sir. Sir, already we are talking about our students. I already have sent one student to your department from my undergraduate college.
Yeah, you
Who is
said
that?
me that day. No? Yeah, it is
One
very
student
good.
from my college. I have sent him to your
What is
department.
the name of the student?
Dibborana Shaharoy. He is doing MSc.
Not in my
Masters.
department.
Okay,
It's in my
okay.
department. Some other
Yes,
department
yes, yes.
maybe.
Yes,
I have one PhD student who, who is from West Bengal, Pushpendu Kundu. He
Okay.
did a lot of work on, the algae, the red seaweeds. Yes. He
Yes,
recently completed his PhD.
Okay, but he is doing MSc, not the PhD program.
Okay. Right.
I hope he will join soon.
Oh yeah.
Thank you,
Right.
sir. Thank you. Very nice.
All
talk.
right. Thank you so much.
Yeah,
Nice
thank
day.
you.
Thank you, sir. Thank you so much for your precious time. Moving on to our next lecture, we have Professor B.B. Chaugule with us. Welcome, sir. Thank you so much for your time. Sir, can you hear us? Chaugule, sir, can you hear us? So you're not audible. Please unmute yourself.
Yeah, I'm doing. Am I audible now? Am I audible now?
Yes,
Yes, sir.
very clear.
Yes, sir. Yes, sir.
Thank you. Thank you.
So before you start, sir, Professor Chaugule, he was formerly in the head of Department of Botany, University of Pune. He discovered new type of life history in red algae, Batrachospermum, a genus included in textbooks all over the world as typical example of haplobiontic life history. This discovery leads to revision in the textbooks, accounts, and classification of red algae. He taught in algology, bryophyte, pteridophyte, angiosperms, and aquatic ecology at general level. His research interests include taxonomy, marine botany, ecology, laboratory and field cultivation, cytology, and life history and application of algae. He has been the head of the department for a long time. He enrichment, excuse me, sorry. And he also did a lot in instrumental in seeking UGC assistant program and grant for infrastructure development. He has been the chairman of Board of Studies in Botany, University of Pune. He also revised undergraduate syllabus to accommodate innovative and skill-oriented courses as computer applications in botany, tissue culture, gardening, mushroom cultivation, seed technology, and others. He has been the principal investigator of several projects under DST, ISRO, DBT, and other works as well. He has been the key investigator in the initial work which led to the establishment of the world's largest facility of microalgae biomass production at Gagwa, Jamnagar by Reliance. He has guided 15 MPhil students and 13 PhD students. He has more than 80 papers, research papers in the area of taxonomy, cytology, life history, ecology, bioremediation, fatty acids, pigments, and pharmaceuticals of algae. He has also been the recipient of the Lifetime Achievement Award by Krishnamurti Institute of Algology in 2016. He has been a member of many eminent scientific societies. He has been participated in cruise participation of RV Gaveshani, Arabian Sea and Indian Ocean, RV Gaveshahi, Lakshadweep Sea. He has visited abroad in UK, Sheffield University. Also, he has been the consultant of the biodiesel from marine microalgae with Reliance Industries. He's a member of review committee on mission mode program on seaweed cultivation and utilization CSIR New Delhi. Also in the establishment of State Marine University of Government of Maharashtra. Thank you so much, sir, for your precious time. Over to you, sir, for your lecture.
Welcome, madam. Thank you very much for nice introduction. And the audience is right from undergraduate, postgraduate, and researchers, isn't it, madam?
Yes, sir. Yes.
So I have devised a lecture in such a way that maybe researchers and teachers might feel that they are familiar, but it is more student oriented. Yeah.
Yes, sir. You please continue, sir.
Yeah, I'll cut.
Share karache?
Share.
Okay, give me a minute. I'll help you. Are you able to see your screen? Are you able to see his screen?
Are you able to? Hello, madam.
Yes, sir. Yes, sir. It is visible. Yes, sir. It is visible, sir.
Okay. Thank you, madam. Thank you. First of all, let me thank you, Professor Ruma Pal for inviting me to on a group of algae which I like much, although I worked on other groups of algae also. And particularly, I am extremely happy because I have been invited. It considered me as a great honor to receive invitation from you all. At least, the function which is concerned with the great Professor Probir Chatterjee. And when I talked, although I was aware about the contributions of Professor Probir Chatterjee, particularly in parasitology and the cyanophyce, but when I talked with Professor Rumapal and Professor Samit Roy, and then it was a great surprise. Of course, I was astonished by the unbelievable credentials that Professor Probir Chatterjee has contributed. Although a person from mycology background, but worked on algae, bryophytes, pteridophytes, and I was told that the person was having a wonderful skill in teaching. The wonderful teacher having a passion in research, and more than that, he was a champion in cryptomic groups. Particularly, I was told that he was equally good in bryophytes, pteridophytes, and people used to contact him for identification of bryophytes, pteridophytes, in addition to algae and fungi. Such a great person, and I really feel very much honored to be associated with this. And more than that, he was a wonderful teacher, I was told. And teaching, of course, was his first priority. It was close to his heart, and this teaching was such a wonderful and marvelous. I was informed that the class was in a mesmerizing mood. Really, I'm very much honored. And now coming, once again, thank you, Professor Rumapal, for inviting me and giving such a great honor. Now coming to the topic, I'll be very fast because I thought I should first introduce red algae first, and then we'll go to biotechnological aspects. Now, when we talk of red algae, we must remember that all red algae are not red in color. You have, of course, red algae are red because they contain additional pigments called phycobilioprotein. And phycobilioprotein, as you all know, contains both phycocyanin, phycoerythrin, and allophycocyanin. And depending upon the amount of phycocyanin, ratio between phycocyanin, phycoerythrin, and allophycocyanin, of course, it depends upon where the alga grows. You have a variation in the color, right from light pink, and then blue-green, and green color also. See, mostly the freshwater and soil red algae would be other than red colored. Let me talk about some of the interesting features, but before that, let me tell you that this is the group is as a group characterized by pigments I told you, but the other character, which is a negative one, that is absence of motile reproductive structures. And in this respect, this particular group is a handicapped one. Handicapped one in the sense being in a water but unable to swim because their reproductive structures are non-flagellated, or on a land cannot walk. But in spite of that, this particular group has followed such a wonderful strategy. I'll talk a little later on about this, because of which this particular group has become the most successful group among the algae in terms of number of species, number of genera, in terms of services given to the nature by this particular group of algae, as well as if we look into the human utilization. And this is something I usually tell students, that don't feel depressed, suppressed. Look at the red algae. the way they have modified their lifestyles and have become a dominant one. So this particularly among the algae, it is one of the inspiring group. In nature, they are the primary producers. Animals graze, aquatic animals, they provide oxygen, enrich the environment, and more than that, they are the carbon sequester. Particularly coralline red algae, where they have an ability to precipitate calcium carbonate on their body, which calcium carbonate is nothing but it is a deposition insoluble in water. That means CO2 is permanently bound there. It cannot be again, comes back to the environment. Thus, they are most responsible for maintaining the carbon cycle in the Earth's environment. Besides this particular group of algae is concerned with human welfare, and initially and also currently also there are many red which are harvested from the wild, but because of the increasing demand, they have been cultivated on a large scale, like algae like Porphyra, Kappa ficus, then Pomaria. Because of food phycocolloids, agar, agarose, carrageenan, and nowadays there's a more importance to the phyco supplements, which are called as biostimulants, soil additives, fertilizers, the semi-liquid fertilizer, which is present either in the form of liquid or powder or granules, and also is used as animal feed. Now, besides, it has a tremendous great potential in the form of pharmaceuticals, nutraceuticals, cosmetics, and in addition, phycoremediation. Now, phycoremediation, I don't know whether I'll get a time to explain or not, but you know that there are, in also West Bengal, you have a large number of shrimp farms, and where you have what is followed is the intensive cultivation. And because of that, waste material accumulates. Only 20, as you know that whatever the feed that is given, only 20% it is utilized, 80% goes into the water. And experts have shown, and now people have been following also in Indonesia, particularly Malaysia and Vietnam, where Gracilaria is grown along with shrimps. So Gracilaria takes all those, uses all that material, waste material, and also gives oxygen, and thus this co-cultivation helps in maintaining the clean environment of shrimp ponds. And there are, as I told you, that this is the upcoming area of the utilization of red algae. Because they have a large number of pharmacological applications like antioxidants, anti-inflammatory, antifungal, antiviral, anti-proliferative activities, contraceptive, wound healing effects they are anti-diabetic activities, agar-agar, for example, hypolipidemic activities, cytotoxic, phytotoxic, and also they have shown to have insecticidal activity also. If we look particularly into the amount of seaweed biomass that is used and the market, it is astonishing. Porphyra, for example, it is one of the food nori students are aware of that, where the production is more than 900,000 wet tons, and the overall turnover is 1.5 billion United States dollars. Carrageenan and agars are important. Particularly, we are aware agar-agar is used in a tissue culture. We are all aware. Carrageenan has a large number of applications, and agar industries, again, you have, the amount of agarophytes that are used is 1.25 thousand, and the trade is $1.32 million. Carrageenan, and which is now carrageenan industry is growing up in India. It was not there previously. With the beginning of this era, slowly Kappa ficus cultivation is going on, and carrageenan industry is now establishing. It's again one of the largest industry, 1.40 thousand dry tons of harvested, it's a dry. So into 90, that makes the wet material, and the trade is $600 million. The other industry which is coming up, and you have wholesale dealers in Calcutta also, and that is related to Phyco supplements, which is about 1.22 million wet tons, and it is $53 million, the overall. Now where these red algae grow? In the structure, morphology, they are diverse. And the other thing, let me clear that many of the students and teachers and researchers think that red algae is a complex group, and many students they put red algae as an option. They don't attend it. It's mostly because of the textbook accounts, which is more complicated. But let me tell you, my experience is red algae is most simplest group, having uniform type of reproduction, uniform type of life cycle. Only there was some controversy, and because they have a 3 phases. Normally you have a gametophyte and tetrasporophyte, or sporophyte in other groups of algae, or only gametophyte. Here you have a 2 phases, and therefore, see, more the things, more the morphological entities, additional terminologies. So all these additional terminologies have made red algae complicated. So let us forget about terminologies. If you look into the morphology and the way things move, it is very uniform. Now let us see where these algae grow. In seawater, they grow in marine environment, they grow in marine environment, particularly in the intertidal region. And in this intertidal region, they are more towards the sea portion rather than towards the land portion, because they are adapted for the deeper water environment. They are more in a lower intertidal zone and infralittoral zone. That means that is the area which is never exposed, and it is because where the light is weak compared to the other area in the intertidal zone, and therefore you have a phycoerythrin which traps the light and gives energy, and these are the red algae which are present in the form of attached to the rocks. Now you have a red algae also in estuarine areas. They grow attached to the rocks, and more than that, tree trunks and nematophores of the mangroves. Sundarbans, for example, it is one of the favorable area for red algae to grow. Now, when would you go for collection of red algae in the marine environment? In freshwater environment, okay, water is not much deep. For those algae which grow on a soil, you can go and pick up. But for marine environment, you have to go when the water recedes during high tide. This area is totally covered. You cannot go and look and collect. So you have to wait till water recedes, and during low tide, water goes back, and high tide it covers. So during low tide, you have to go. Again, in our country, you have 2 types of tides, mixed diurnal tides. That means every day you have 2 high tides and 2 low tides. And the water that recedes or water that rises again varies. So the best time for collection of the algae, marine algae, is a time when sun, moon, and earth are one in line. That means either new moon day or no moon day, where the high tides and therefore low tides are also better. Water resists much more area of the intertidal zone is exposed, and so you can go and easily pick up the material. Whereas on other days, when the moon makes some angle with the earth rather than in one line, the tides are not good. That's not a good time for collections. Now, I in the beginning I told you that red algae is the most successful group. You have their representatives in marine water. You have their representatives in estuarine areas. They grow in a fast flowing streams where others cannot survive, others cannot attach, where the spores would get carried away, but red algae manage to survive. They grow in ponds, they grow in rivers, and also they grow on a soil. Can you see the red pigment, which gives a contrast with other groups of the algae, green, blue, green, and red. Now coming back to the terminologies and what makes the red algae most successful groups. As I told you that whether it's a female reproductive structure or male reproductive structures, both are non-motile. Female reproductive structure is attached. Male, although they are liberated, there are no flagella. So this only it is the water current, that means chance, that means luck, will take to the trichogyne and would take part into the fertilization. Therefore, one of the strategies that is adapted is to produce large number of male reproductive structures, spermatangia. Now, the second adaptation is the most important adaptation, that once the zygote is formed, from the zygote, then many filaments arise. Forget about the terminologies. Branch filaments arises, and at their tips, then carpospores are produced. Carpospores are nothing but zygote-like spores in all respect, except the position. So, in other words, with the single act of fertilization, the strategy here is, although they are handicapped, strategy is with the act of single fertilization, large number of fertilized products in the form of carpospores are produced, thus giving more advantage to the other groups. So even though many of the carpogonia female reproductive structures remain unfertilized, because of this additional phase, that is cystocarp, carposporophyte, zygote-like large number of spores will be produced. So here in this lower group of algae, you have only one cystocarp coming at the point where carpogonium is present. Whereas in higher groups, look at here, in gigateneals, cryptonemeals, where after fertilization, a tube arises that moves and meets with a cell which is rich in nutrients, and their cystocarp will be produced. Again, one of the tube will arise here, particularly this figure, particularly figure number E. Another tube arises that seeks that looks for the cell which is rich in nutrients, which we call as auxiliary cell. Then another cystocarp will be produced. Thus, from single fertilized zygote, up to 40 cystocarps can be produced. Then imagine the number of fertilized products will be produced. This large number of fertilized products, although group is handicapped, it has become most successful. And since this cystocarp is a diploid and that is present on the gametophyte, so terminology is that you have a presence of haploid gametophyte, diploid cystocarp, and then tetrasporophyte, and tetrasporangia are the sites of meiosis. Now, all these features are used in a classification of red algae. Now, this is about red algae also occur on the soil. This is Porphyridium growing in one of the greenhouse near Pune. It's pink in color. You have a Porphyridium species which are also blue-green in color. Here, phycocyanin is in abundance. In this case, the phycoerythrin is in abundance. Now let me take very briefly the survey of the diversity of the red algae, filamentous, then the thalloid, typical parenchymatous one, Porphyra, and during monsoon season, all our rocky shores, particularly on western coast, will be full of Porphyra plants. Then this is the freshwater alga, Kamsupogon, that has been reported by Professor Rumapal from Bengal also. This is a filamentous alga. Then another Kamsupogonopsis, which is also filamentous. Then now these are the previous members are from the Bangles. Now we come to the higher group, which is Floridophyceae. And if you have a fish aquaria in your house, look for such a branched blue-green algal growth. It is a red alga called Audionella, which is filamentous. Then, and here, getting rid of this Audionella is really a problem. That's another business people have been doing, the removal of the algae by using algicides. Then this is a Batrachospermum. Everyone is aware of it, monaliform, which reproductive structure is cystocarp. And then coming to another freshwater alga, the Batrachospermum is freshwater. Another freshwater alga is a Lemania, which grows in a Manipur. And this alga grows attached to the rocks. In winter, it grows in a fast-flowing water river Chipki, and this alga is edible. I'll talk a little later on about that. Then other members of the Nemallids like Asparagopsis, Helminthocladia, Ligora, Cinea. Then Giladials, you are familiar with the Giladium, which is a source of agar. Then these are the members of Cryptonemales, another order. You have a thalloid and other Cryptonemales, which I referred regarding the carbon sequestration. These are the algae which have ability to precipitate carbon, calcium carbonate on their body. And this is a calcified alga called Melobacia, which form crust on the rocks. So see, since this calcium carbonate is insoluble in water, carbon dioxide, carbon in the form of carbon dioxide, in the form of calcium carbonate is permanently lost. Then this is another group called Gagatinales, and these are the cistocasts which are seen. And this is a Gracilaria, which is a major source of agar in the world. Then this another alga which is common on our coast is a Hypnea. This is the used for carrageen production. Then the next beautiful group of algae, order Lamina, order Rhodymenias, to which Champia belongs, and these are the cistocasts fruiting bodies. So plant is gametophyte, and these bodies are diploid one. Then this is the last group, Ceramales, Polysiphonia. Students are probably familiar with this. You have a reproductive structures in the form of spermatangia, female reproductive structures, and diploid tetrasporangia. Now coming to their utilization. If anybody wants to know more, yeah, they can interrupt my talk and they can ask me also. Now, this is a photograph from Chipki River. See, this alga is used traditionally. So it's for centuries. It is collected, dried, and sold into the markets in all over the Manipur, particularly in Imphal. This is a photograph from the Imphal market. Now, this alga has ethnopharmacological uses, and particularly is used as an abortive measure by the local people. Then it is also used as an aphrodisiac, then a diasic, and then also if anybody is suffering by kidney stone, okay, this alga is used, and it is said that within 10 days the problem would be resolved. And the filaments of this alga are roasted on a fire and used to treat the diabetes. More than that, what we studied, we surveyed and we studied the fatty acid profile, and we realized that is almost 24.5% of the lipid content contains EPA, which is very, very useful. Particularly, it is concerned with the development of brain, and therefore it is a very important one. And in this alga, you have, there is an article by Dept, D-E-B, Dept. The person was from Botanical Survey of India with a geology background. And he was, as a part of his geological work, he was surveying, and when he analyzed Lemania, he found that this Lemania contains silver, and you know that how valuable silver is also. So eating Lemania, the people also get some silver. Now coming to the alga, which has been reported by many from West Bengal.
I think there is some technical glitch from Chaugule sir's side. Sir, you're not audible. Sir?
কোনোভাবে নেট কানেকশনটা ছেড়ে গেছে মনে হচ্ছে।
হ্যাঁ, স্যারের দিক থেকে।
ঠিক।
আচ্ছা, আমি ওনাকে ফোন করছি। Sorry for the technical glitches. Please give us some time. Sir, actually, you're not audible. Your connection has gone. If you can join back again. Yeah, he's no more in the link. Okay. So he's trying to join again. So just few minutes. Sorry for the interruption. As you know, virtual platform, you will have these technical glitches. He's trying to join, actually. I called him. He's joining back, so give us little time.
যে বছর লেকচার দিচ্ছেন পুনা থেকে তার নেক্সট
He's joining within a minute, he's saying. There's some connectivity issue from his side. He's joining.
Hello, hello.
Yes, sir. Yes, sir. Yes, sir.
Yes.
Sir, please share your screen. Yes, sir. We can see you. Yeah, yeah. Thank you, sir. Please continue.
I'm sorry.
He's not able to. I think you need to just enable the screen sharing for him.
Yeah, it's there. It's there. He's the oh. Yeah, I'm doing it.
Yeah. Okay. Yeah, that helps. Thank you.
Yeah.
Hello, hello.
Yes, sir. Yeah, you're audible, sir.
I'm
Audible
sorry.
and visible.
Yeah. Kindly accept my apologies because of the
It's okay,
internet.
sir. We all know it's technical thing, so nothing from your side. Continue, sir, please.
Right. Where I was near Catenella, isn't it?
Yeah, yeah.
When the connectivity was lost. See, Catenella is the alga which is common in the mangrove areas, and that has been reported by many, including Professor Uma Paul, and it has been worked for its protein, carbohydrate, lipid, and astaxanthin content, indicating that it can be used as a food and feed. And astaxanthin also, presence of astaxanthin indicates that it can be used as an antioxidant agent. Because of most interesting thing is this alga is used, is eaten, consumed in Myanmar, in Burma, And because of that, there the cultivation strategy has been followed. It's a very simple technique where bamboo sticks are inserted in the mud flats where Catenella and other Caloclasa grows, spores attach, and they grow such a luxuriantly on the bamboo poles. And there are also attempts, biotechnological attempts to grow spores and obtain the seedlings. I mean, it's essentially to get the propagules. Now, as I mentioned, Catenella in Burma, again, it is whatever that is used as a food and sold in a market that comes from the wild harvest. And in many places, this alga is combined with enteromorpha and sheets are prepared, and these sheets are sold into the market. The other market, besides food, other market is a pigment. Pigments, as I told you, porphyra and all red algae, they have a phycoerythrin, and currently phycoerythrin is extracted from red algae, and porphyridum is a major source. Now, phycoerythrin has a different coloration, depends upon the place where this alga grows. Now, this particular, the carrageenan, is from Kappa ficus. So you have a Kappa ficus which is used for carrageenan mainly, then for pigments, and scientists from CSM Sierra Bhavnagar, they have developed a technology where Kappa ficus can also be used to obtain biostimulant. So before extraction of carrageenan, the sap is obtained, and that is used as a biostimulant. And these are, porphyridum is most important, and therefore grown on a large scale, having various uses as strength immunity, pharmacological, medicinal uses, then in cosmetic industry, and fatty acid in the nutrients, and powder is used as a feed and fertilizer. Now, because of this, see, look at this particularly I included, look at the different coloration that phycobilins from red algae shows, such a wonderful, beautiful colors it shows. And because of that, you have a cultivation on a large scale, and this is a raceway pond. And you have a variety of cultivations, raceway ponds, then tubular ponds, covered raceway ponds, then erected ponds. And now since there are problem in harvesting porphyridium secretes the exopolysaccharide, and therefore there are problems. Whenever you want to harvest it, you have to collect the water sample with porphyridium. So now a new technology that has come where it's a stationary, porphyra remains stationary. So when the water is collected, no material would be wasted in this case. Again, pigments. See, these are some of the slides I included only to show the viewers, particularly students, that it's a reality that large number of industry people, they are in a market and they are selling it. Now, because of this importance, a lot of biotechnological work has been done and being carried on porphyridium. And the first step, before going to biotechnology, first study is morphology. It's a classical study which follows physiological, then biochemical, and then the other molecular studies. Genome of this particular red alga, porphyridium, has been completely studied almost 10 years back, we know, and therefore now it is possible to manipulate the different metabolic pathways. Now coming to the other alga which is cultivated in our country and elsewhere outside also on a large scale is Kappa ficus. Basically, it is the technique that was developed and by the scientists of Central Salt Marine Research Chemicals, Bhavnagar. And more than that, in Pamban region and in Gujarat also, but more in state of Tamil Nadu, southern portion of the Tamil Nadu, where women, women, they have formed self-help groups, And each of the self-help groups now of the women, they are in this particular business of growing Kappa ficus. And now each family, because of this, is earning more than 15,000 rupees per month. So this is one of the green area for work. Now this, as I told you, Kappa ficus has two types, one pink colored and green colored. And again, but there are certain problems with Kappa ficus, particularly in India. Kappa ficus that we have been growing is a vegetative one without any reproductive structures. And okay, anyway, there are not much problem because cultivation has very recently started. But sooner or later, because it is the same vegetative material that is used, alga will lose its vigor. It will take more time for its growth. Percentage of carrageenan also will go down. Plus, there are also chances of growth of the epiphytes, fowlers, and also there are chances of the attack by the bacteria causing disease called as ISIS. Luckily, we don't have these, but in Philippines, Indonesia, this is the alga which is grown on a very, very large scale. Philippines, Malaysia, and Indonesia are the great players in this particular. So because of its vegetative nature, you can't use these explants and again to grow. And therefore, you have here biotechnological role. It's a tissue culture. It's a schematic diagram where the explant is allowed to grow to form a callus. And from the callus, then the propagules, younger plants. There is a paper very recently published, only a few years, 1 year back, by a scientist of Vietnam, and they obtain a callus. Here, the explant that is used is a small piece of the thallus, middle portion and upper portion. And using different media, you have a development of the callus at one end, both ends, or in certain media and environmental condition, depending upon the health of the callus. Then callus grows, and once after 2 months, the filaments. See, the problem in red algae, the higher red algae, including Kappa ficus, Gracilaria, Catenella, they are not true parenchymatous. They are pseudoparenchymatous. So the water tissue used, the term tissue is not correct one. These actually filamentous. These filaments, after 2 months, start accumulating pigment into them. And once they accumulate the pigments, then they slowly growth occurs, and there forms what are called as somatic embryos. Now, term embryo, technically speaking, is not correct used for angiosols, but anyway, for the purpose of conveying the message, let me use terms embryos. So somatic embryos are formed, and once the somatic embryos grow, and once the somatic embryos are transferred into liquid medium, plantlets are formed. So with this tissue culture technique, now it is possible to get the plantlets in a large quantity, and also by using genetic engineering methods, one can also improve the strains to have a greater vigor, to have a more carrageenan, to have a more pigment, and to disease resistant. Porphyra is another one. There's no cultivation in our country, but again, it's one of the major industry. And here, the propagules are produced by using the part of the thallus. Those are cut. Once those are cut, from one end, rhizoids arise, and from other end, for example, look at here, from other end, thallus grows. Thus, from single thallus, large number of propagules can be produced. Now, coming to Gracilaria, very specifically, I have included this particular book in my talk. Actually, Gracilaria cultivation program was initiated in 1989 for the benefit of the countries and states which are in a Bay of Bengal region, like India, then Myanmar, Malaysia, and so on. And whoever is interested in knowing the methodology of cultivation can refer this book. This book is available online. Now, Gracilaria has a variety of uses, like agar, lipids, fatty acids, steroids. I'll not go into the details here. But we have a Gracilaria cultivation undergoing, but not the Gracilaria which is used for the production of agar, edible agar. Here we are depending upon the wild harvest, particularly in the region of Bay of Park Bay or the Kursara Islands, where the Professor Rumapal referred in the beginning. Now, Gracilaria dura has been recommended for cultivation in Indian waters. Why Gracilaria dura? Because in Gracilaria dura, you have a more percentage of agarose having a better quality. And you know that agarose has a large number of utilization in a molecular biology studies. So Gracilaria dura, and this is the life cycle of Gracilaria. The ways to improve is to get, number one, in a previous case, as I told you, to get a vegetative thallus, cut it and get. And number two, use spores, either tetraspores or carpospores to get more and more sporelings. The other way that has invented by the group headed by Dr. Vaibhav Mantri from Central Salt Research Chemicals, where the plant thallus was cut into 2 centimeter pieces, and then they were placed in a medium where the growth promoting substances were added. Within few days, 15 days, the embryo-like structures are formed, which can be used as the explants. See, in Kappa ficus, as I told, there's another problem I forgot to mention in Kappa ficus. See, Kappa ficus is very sensitive to salinity, where if the salinity goes down, Kappa ficus dies. And that is another major constraint on promoting Kappa ficus cultivation on entire coast of India, because you know that because monsoon seasons where the salinity goes down. So one of the major problems that Kappa ficus industry in India, cultivation project in India is suffering is lack of starting material. And therefore, whoever comes with some solution will have a big name as well as also big money also. Now this particular, hope I'll take another few minutes. Now, although the topic allotted to me is the red algae, but with kind permission of the organizers, I like to talk on other groups of algae because it is something concerned with your state, Bay of Bengal. Now, this is a photograph, and the news appeared 2 years back. Here is the lady who is, we got fed up because rice cultivation was in a danger because of the encroachment of the seawater, number one. And therefore, the men have to move to the cities, and ladies, because they have no other alternative except to collect bee, honey, and all those things. And also, the risk in going into the mangrove forest and collecting the honey and other forest products. So there's another very, very marvelous and new way that has been now provided, where algae is not a red alga, the algae is collected, which is growing in nature. Also, ponds are developed where the seawater accumulates and algae is allowed to grow. This is also used. This is dried in front of their house, and this dried mass is used as a fertilizer, as a manure, and thus the ladies who are really, they were in a financial problems. Now they are getting some relief in the form of seaweed collection and crude cultivation in ponds. Thank you very much for giving me a chance to interact with you, and also as I mentioned in the beginning, to be It's a very, very great honor for me to be associated with a program in the honor of Professor Probir Chatterjee. Thank you very much, madam.
Thank you, sir. Thank you so much. So the session is open for interaction. Participants, if you have any questions, you can please go ahead. Even the chat box is also open. So please, participants, if any participants have any questions.
May I start with a question?
Yes,
Yeah.
absolutely, sir. Please go ahead. Professor Shomit,
Chaugule,
sir.
thank you so much for this brilliant lecture, particularly on the various aspects of culturing, which is such a big aspect of our present economy. But I had one theoretical question regarding which will you support? How many classes under the division Rhodophyta?
Oh, okay. That is one of the interesting question. See, the problem with algae is, it's a right question also. And that is one of the reasons why students keep also red algae, why I mean the option. See, if you follow classification by Fritsch, you have Bangials, the Nymalials, Cryptonemials, Giladials, Gagatinales, Rudiminals, and Ceramils, 7 groups. Now, because of the advancement in molecular biological tools, number of classes have now on a rising scale. Now, let me give example of Nymalials, in which Batrachosperma was included as a Nymalials, Batrachospermaceae and Batrachosperma, you know. Now, Batrachosperma has become order Batrachospermales. You have but genus Batrachospermum now with the molecular biology, phylogeny technique, Batrachospermum is now considered to have 8 genera. So this taxonomy is changing. You don't have now fixed number of orders. It's rising and rising.
No, my question was how many classes? Previously, the general concept was that Rhodophyta, Rhodophyceae, and then Bangioideae and Floridiophyceae.
Okay.
Now,
No, I spoke about the order. Sorry, your question is on classes. See, classes 2, Bangiophyceae and Floridiophyceae.
Bangiophyceae and
Floridiophyceae. Primitive
2
group,
classes.
Yeah, 2 classes. Primitive group is a Bangiophyceae, and advanced one are in a Floridiophyceae. Those with unicellular and true parenchymatous are in Bangiophyceae. Okay. And those filamentous and pseudoparenchymatous all are on Floridiophyceae. Besides, they are characterized by presence of pit connections, where a Bangiophyceae, they lack pit connections.
Right.
Then reproductive structures are very simple in Bangiophyceae. Either majority of them are asexual, and Porphyra and others, where you don't have a complex type of, there are no branches, carpal branches or spermatogenesis, the vestigial cell that is converted into male and reproductive structures. And carpospores are directly, zygote divides into 4, 8 cells or 16 cells, and there are no gonimoblast filaments into. In other words, there are no post-fertilization changes in Bangiophyceae.
Yes. Thank you so much.
Welcome, sir.
Thank you, sir. Thank you, Shomit, sir. Now we have a question from the participant, Munju Shri. She has asked, what is the salinity range of Kappa ficus?
3.5 to 4.
Okay.
It can tolerate higher salinity, but cannot tolerate low salinity.
Okay, I hope she's got the answer. Thank you so much, sir. Just now I had a talk with Professor Rumapal. Actually, due to technical glitches, she's not able to connect. She's not able to talk. So she's there, but she's not able to connect due to some technical problem from her side. So she has sent a lot of regards to you, sir, from Ruma ma'am. And thank you so much for taking out your precious time from your busy schedule and enlightening us and the students and the participants with your information. Achha, we have another question over here.
Oh,
So
good,
sorry
good.
to interrupt. Sorry
No,
to
no,
interrupt.
Good,
It is asked by the same participant. Is there any changes in distribution due to anthropogenic pressure?
Yeah. Because of anthropogenic pressure, say, particularly eutrophication, we are losing many algae. I mean, including red algae also, both fresh water as well as marine water. Yes. And the very interesting thing in our country is not much survey. I mean, not much work has been done on a taxonomic basis. And because of this anthropogenic pressure, particularly urbanization and tourist activities, we are losing large number of precious gene pool. And the problem is because of the low discoveries, we do not know even what. I mean, there are many species that are lost even before their discovery. It's a very pity situation in our country.
Thank you, sir. I hope she's answered. So she's asked another thing. Is that acting as an indicator species? Isn't it is an indicator species or not?
Oh, yeah. Presence and absence. Yeah, these are really good indicator of the water quality. But the problem is, if you compare with other groups of algae like green algae and blue-green algae, cyanobacteria, red algae distribution-wise occur in a few water bodies, and those are not commonly present in water bodies, freshwater bodies, and therefore, I mean, those cannot be universally used as an indicator of species. But where those have been recorded before, yeah, for determining their quality, those can be used. But you cannot compare their value with the green algae, particularly members of chlorococcales, then diatoms and cyanophyces.
Yeah, I think ma'am has been able to connect visibly, but ma'am is not audible.
Oh,
ma'am, you're not audible. You're unmute, but still you're not audible. We can see you, ma'am. As you know, these technical things, yes, sir, ma'am is kind of waving at you if you can see her.
Yeah, I can see her. Yeah, right.
She has sent a lot of regards to you for your time and for your informative and knowledgeable presentations. And I hope the students and the participants have been enlightened. So thank you so much, sir, from our heart. Yes, sir. Thank you so much.
Yeah, one sentence from me.
Yes, sir, absolutely. Yes, sir.
Thank you all for your patience hearing, and also please excuse me because I talked very fast because I wanted to cover everything in a shorter time.
No, sir, not absolutely. We have all understood. I hope all the participants actually.
Yeah, all the participants. But if anybody has any query, certainly they can contact me anytime.
Yes,
I'll be really happy. Because absolutely. Our major issue is to serve. We have to see that phycology survives, and phycologists also should survive. That is the major issue now.
Yes, sir. Thank you once again, sir. So with this, we come to the end of this technical session. With this, we will go to the lunch break and we will meet again. Thank you. We will meet again at 2 p.m. for the technical session. That's the poster session. And then we have another lecture from our international speaker, Professor Andrew from Poland. And so meet you again at 2 p.m. Till then, have a lunch break. Enjoy your lunch. Thank you. Thank you so much.
Thank you.
Participants can leave. We will join back at 2 p.m.